Welcome everyone to our bioelectrodynamics webinar. I would like to welcome all the attendees and our speaker today. Before we start, I should announce that our webinar is supported by the International Union of Radioscience, as well as by IEEE, particularly Czechoslovakia section and the Engineering in Medicine and Biology chapter. I would like to introduce our today's speaker, Dr. Arunas Stirke, and I would like to say that he's currently both postdoc fellow at the Laboratory of Micro and Nano Devices, Institute of Solid State Physics, University of Latvia, but also he's a senior researcher and head of Bioelectrics Laboratory of Functional Materials and Electronics, Center of Physical and Technological Sciences in Lithuania. He's Doctor of Chemical Sciences. He was been working already since some time on pulsed electric field applications on yeast, and also he's member of International Society of Electroporation Technologies and Base Therapies, and a board member of Lithuanian Society of Biophysics. And before further ado, I would like to give floor to you, Arunas. Please go ahead.
Okay, hello everybody. So I will share my screen, and I hope it's fine.
Yes,
yep.
Perfect.
Great. So thank you, Michael, for introduction and for great opportunity to represent my work. Of course, it will be as a part of my work, which I'm doing as a postdoc researcher, and I'm senior researcher in Lithuania and Latvia, in both countries, in both institutions at the same time. Reason is that because I am working in the Institute of Solid State Physics, University of Latvia, to develop some specific devices for my work, and I am as a postdoc researcher in micro and nano device laboratory. But now I'm speaking from Vilnius, which is a really nice and amazing place, not just to have fun, but to have really great science, because we have a new facility to do that, and it is like as a valley of science, which can be seen a little bit here. So it's our building here, FTMC, it's Center for Physical Science and Technology. Here it's a Vilnius University Life Science Center. Here it's a big library, and the whole area here it's dedicated for various universities and some universities, some departments of Vilnius universities. So in FTMC, it's quite a big institution. Here working around 300 scientists, around 17 departments in which we are working. It's the Department of Functional Material and Electronics. And as a center, we have a lot of space, not just to have fun, but to have a really good science, because it's the center is equipped by new equipment and really good. I think it's really good to have to have everything in one space, starting from physics and ending till the biomaterials. Of course, in our center, we have open spots annually for physics, chemistry, and material engineering for PhD students. So keep in mind, if somebody wants to, you are welcome. So in FTMC, I am a senior researcher and lab manager of Bioelectrics Laboratory, which is a quite young and interdisciplinary group of scientists investigating pulse electric field effects on intracellular signaling and biological nanostructures to develop innovative technology and prototypes for modern food processing and healthcare. So in general, I really want to or just to tailor some part of our lab work here. And I really want to talk mainly about the proteins. And the proteins for us, it's very interesting, and especially we can split our interest into two different, let's say, levels. As I told here, cellular level and molecular level. What this means, actually, it is very interesting how a PEF-induced effect of proteins can be seen or investigated in cell. So it's a biological level, or how these biomolecules, especially proteins, can act after a PEF treatment in a liquid, or we can say biochemical level. So let's talk about the cellular level, or mainly how the cells after a PEF treatment can act to such kind of abiotic treatment. And it was published year ago, almost 2 years ago, 1 hour article, pulse electric, how in which we investigate pulse electric field effects on expression of some reporter genes, which was controlled by some specific promoter. If you look a little bit into the detail of his research, actually we make some specific cells. How it's called cell transfection. It means we put some foreign gene inside the cells by using chemical treatment, and then we stimulate these cells by pulsing. This pulse was selected as a regular pulsing or as a regular protocol for electroporation. And then we investigate how these cells, does these cells actually feel such kind of treatment. And as I told, the cells was transfected by the plasmids, which holds some specific promoter area and some specific gene reporter. So it's mean if after such kind of stimulation, the cells in a cell, some specific intracellular signals can be triggered, we will see some expression or because the product of this reporter gene is some specific protein which can be secreted out from the cells, and this protein is a specific protein, alkali phosphatase, and then we can see some reaction in a surrounded in a suspension. It means without the cells. And what we got from this result, from this investigation, that it's really works. So it's mean that nuclear factor kappa beta, it's quite pleiotropic factor. It's mean it's react to a lot of stimulus, starting from heat, UV light, and other abiotic treatments. So in this research, we show that pulse electric field act as a abiotic treatment for cells, and it can be used to and this nuclear factor kappa beta promoter can be used as a promoter for to investigate some biological effects of pulse electric field treatment. And it was quite interesting thing that, for example, we worked with I think 6 different kind of cells, and what we saw that there is some quite big difference when we are using cancer cells and the normal cells. So as you can see here, the expression rate was two times bigger comparing with a control. But if you look into the normal cells, almost nothing in an expression rate. So it's mean that such kind of system not only it can be explored as an instrument not for all cells. And in this investigation, we understand that if you are working with regular, let's say, micro or millisecond pulse electric field treatment, there is a lot of other factors can be other factors that mean that after such kind of treatment, the effects of this such kind of pulse is very, very broad. So it's mean you will get not only the electroporation as it is, but as it quite normal in such kind of protocols. But we saw that it's really quite such kind of a treatment by using microseconds, producing a lot of reactive oxygen species, which can be as a trigger for nuclear factor kappa beta promoter. So of course we are thinking and trying to build up a system which can be used for nanosecond pulse electric field treatment, as it is let's say much way better treatment, especially for some intracellular signals. And of course, as you can see from this drawing, it's yeah, still a lot of questions, a lot of factors and other compounds can be generated after such kind of nanosecond pulse electric field treatment, but still it's we believe that it could be more precise way to treat the cells and to trigger some intracellular signals. But it's the main problem here. If we want to start dealing with such kind of a treatment with nanosecond electrical pulses, we need some specific treatment chambers. So it is a reason why I start my postdoc project. Of this project, I really want to develop PDMS-free microfluidic chip, which can be suitable for detection and evaluation of nanosecond electrical field treatment, and that kind of treatment would be great to explore as a precise gene expression inducing signal. So is the question why it's PDMS-free? There is mainly two main advantages or disadvantages of PDMS. But PDMS, it's really very nice and quite easy producible material, but it's absorbs small molecules, which is not good for some reason, especially when we are dealing with or we want to treat the cells inside the chip, and of course a gas permeability. So it's mean that in one case it's fine, because if it's gas permeable, you can use such kind of PDMS chips to grow the cells very easy, because for cells it's CO2 level and some specific composition of air is required. But in some cases, if you want to grow some specific cells which we are not allowed to grow in or you want to maintain some specific air condition in a chamber, then you need to develop some specific PDMS-free chip. In my design, it looks quite simple, but yeah, we are working mainly with We've had 3 main techniques, starting from stereolithography, soft lithography, and CNC milling. It can be done quite easy, but there is a lot of areas which could be, or areas in which we are still struggling. And if you look into the cross section of our chip, it's nothing special. Just like actually, it's a COC slide. So you see, it's a specific plastic. It's not a glass. It's a plastic. And here we are using some specific dual curable polymer to form some space for cells. And between these 2 polymer layer, we are placing a porous polycarbonate membrane, which is a base or substrate for our cells to grow there. And here you can see a chamber. It's quite small chamber, but it's quite enough for our cells. But still, of course, to grow the cells, so it's mean that it's not it's a big challenge for us and for who is developing such kind of chips, because you need to understand how the cells are growing there, because as I told you, we are placing these cells on a polycarbonate membrane, which is not very biocompatible. Yes, it's compatible, but still we need to enhance that or to grow or to seed on such kind of a substrate, and we need to. We are still developing the protocols for how to attach these cells on this polycarbonate membrane. It's more or less, it's everything, it's already done. Yeah, we have a protocol, and still to seed the cells, it's a one challenge, but there's another, to understand how we are growing there, how we are alive or dead. Did we form some layer on it or not? So we need to develop some specific sensing setups. And here you can see a pH sensing setup, which we already developed, and it's yeah, I don't want to go into the details, but it's looks like as a small spectrophotometer on a chip, which can be used to measure pH changes in a channel. Another thing which is very important. Okay, so here it's pH is sensing some electronical circuits, which is, I don't think it's too much interesting for you, but it can be done in our center, because as you already know, I'm working in a functional material and electronics department. So we have quite a good team, which we are mainly working with electronics and devices. So it's really good to have such kind of opportunity to not just to buy or to integrate some commercial available sensing systems, but to develop by us. So another parameter which is very important to understand or to maintain the cell growth inside a chip, it's the oxygen level. So for that, it's from a beginning, it looks very easy to do that, but in reality, it's not easy, because the oxygen, it's everywhere. So and for such kind of a measuring system, if you want to measure such kind of a concentration of oxygen in a bottle, which holds a medium for cells, it's fine, but it's really quite hard to maintain the how to say, to maintain or to prevent the leakage of oxygen into the system. So there's a reason why I'm decided to develop such kind of oxygen sensing setup, which is based on some commercially available oxygen sensor spots. And yeah, we are still developing, but I hope that this week it will be some results. Yeah, so it's something miss in my slide. Yeah, yeah, sorry for this. Where is okay? Somehow it's yeah. Going to the details, there is a third parameter which is very important for my chip. It's electrodes. It's electrodes for not only treatment for the cells which we are growing inside the chip, but to use these electrodes as a transepithelium resistivity measuring system. So I really, so in this case, we need to develop the same electrode will be used for both PEF treatment and for tear measurement. So for that, we need to, how to say, to model or before to understand or to have some imagination how a pulse electric field can be or how the electric field distribution inside the channel looks like, because it's depend from a distance between electrode and the distance between the two COC slides. Actually, these electrodes are sputtered on the COC slides from one side and another side. So, and such in such scenario, it will form some area, let's say treatment chamber. But as you already know that in the middle of this channel, in the middle of this chamber, there is a porous membrane, which can be as a barrier for pulse electric field distribution. And from model, what we get, we get very interesting result that such kind of a structure can be used as a how to say, as the amplifier of electric field, because if we look here, if when the pulse is applied, the pulse electric field goes from like, or electric field is distributed here, and the electric field, if we just take how to say, if we will take like picture, because most of a polycarbonate membrane is an isolator, so it's mean that most of the electric field will be concentrated and form such kind of nice peaks through the hole or through the pore inside of the polycarbonate membrane. It's good, we'll see, but that it's good to know, because when we start dealing with mammalian cells, we really need to keep in mind such kind of effects, because we don't want to kill them. Yeah, if we will just take a look how electric field distribution looks like into or inside of or across the porous membrane, it looks like as a lot of spikes going through the whole area of a treatment chamber. So, and yeah, going into real things, not models. Yeah, the chip looks like. Actually, here there is no yet electrodes, but still, it's the system looks like this. It's quite small device. Of course, there is a lot of stuff around it. It's sometimes much bigger when it's the device looks like, and yeah. So more or less, I'm finished about the biological effects. So it's mean how the pulse electric field can trigger some biological effects. And now I'm switching to the molecular level, which is quite, let's say, new in our lab. And we have a PhD student, Ahmed Taha, who is mainly working with or investigating pulse electric field effects on structural and techno-functional properties of proteins. And we already published one review article with Taha, and we have a collaboration with Danish Technical University, with Federico Casanova, who is a leader of a lab here, there. And what we understand from this review, that it's, that PEF can affect the structural properties. Of course, in that review, we are mainly focused on the milk protein, but still can be, such kind of a knowledge can be explored to understand other proteins. So the PEF, what he can do, for example, for casein protein, he displays a charge, which is, it's good or not, we will see quite soon, because Taha now it's working on the casein protein, but still it's changing. Yeah, and there is, for example, for V protein, which is a component of the milk proteins, the PEF can be used or the PEF can polarize the protein molecules and form free radicals, which can be cause unfolding such kind of proteins. And of course, the unfolding can cause some aggregation, which is sometimes good, sometimes not. Yeah, but let's go to some details a little bit. Now we are working with some bovine serum albumin and starch. How the PEF electric field treatment cause bovine serum albumin and starch interaction, and how its treatment can cause the emulsifying properties of such kind of conjugates. The principle is quite simple. Yeah, we have some mixture of such kind of biological molecules, and we PEF them, and then we was analyzing how bovine serum albumin and starch conjugates or interaction changes after such a treatment, and how this conjugates or such conjugates emulsifying properties changing after such treatment. Still, I really want to emphasize that we used quite mild treatment condition. If you are familiar with some kind of really hard treatment condition, which was used in, for example, there is one webinar from your activity. Professor Sono from Kumamoto University, he is exploring quite hard and quite high electrical field treatment to see or to change some properties of a protein. We are using quite mild, so just few kilowatts per centimeter with 50 microsecond pulse duration. And what we get from these treatments, that such kind of conjugates after such treatment, it's changing some physical chemical processes properties. Let's say grafting degree, and what is very important, the surface hydrophobicity, it's changing. not too much, but still it's changing, and the particle size are changing, solubility is changing, and it's really interesting because we can explore such kind of a treatment for food science, let's say, and can be used to make the emulsion much better. It's mean to enhance or to maintain better emulsifying properties of protein and polysaccharide conjugates, because it's doing, or it's causing smaller particle size, so enhance solubility of the proteins, and I think it could be a good way to improve the emulsion properties of a solution, and can be explored in some food stuff. These results are already published in Innovative Food Science and Emerging Technology. So for whom is more interesting, more details, please go. It's still for free. So yeah, I really want to go to a conclusion. Conclusion, it's looks like as a or from these results, I extract the main thing or to take home message, but to explore the nanosecond electrical pulses, we need to, doesn't matter for cellular or molecular levels investigation, we need to develop microfluidic treatment chambers, keeping in mind that some kind of electrical field focusing structures, let's say, focusing, I really want to use such kind of, let's say, electrical field focusing structures in a microfluidic chamber in order to enhance electrical field or to focus electric field in some particular areas, because as you already know, to have or to get wide high electric field, it's not easy, especially when we want to go to a very short non-electrical pulse electric field treatment. So in that case, we need to keep in mind some technical or design issues can be used to have better conditions. So I really thankful for my colleagues from Latvian Solid State Physics Institute, especially for Gatis Mazalevskis, who is a mentor there, and our of course, my lab in FTMC, bioelectrics laboratory, mainly Neringa Bakute, who is a PhD student. She's working mainly with microfluidic devices. Arunas Stirke, who is a COMSOL guy, who is modeling and doing a lot of helping us to work with devices. Martina Shapurovas and Wojciech Stankiewicz, we are leader for and developing some electronical devices for us, and of course Justina Kavaliauskaitė. And yeah, I really want to. It's a specific, a special thanks going to Povilas, who was, I'm saying who was, because he's going to the United States for 1 year postdoc, and he's still in one of the bioelectrics laboratory leader, and he's working with the yeast as a model cells for responses to pulse electric field treatment. And I really want to show you the video of which was who was a winner for overall and biology category for some specific contest, which were organized by the science journal, Dance Your PhD. Let's gonna look. I hope it works. Yeah. Before okay, let's look.
Everybody has a dream to pursue in a game, find a friend, buy a favorite shampoo to evolve, see progress and never things to do. Once major enough, they could form a crew, feel walk and talk together, be a dream to like no ever, share their joy, being in love, fit together like a glove. Create a new enzyme, flavor, color, pigment, delay, sever hormone toxin in the brain, pharmaceutical cocktail. Once they sense a tingling feeling, motivation grows, no ceiling, no one now can make them stop. Reaching goals will lead to the top. Stimulation makes them sweat. Breathing and living raised by get. No side effects, and don't forget, tasty goodness, fresh mindset.
Even though you cannot see me, I'll be so close you can't resist. Tell me your needs by charging, please. The lightning strikes, now make a wish. Even though you cannot see me, I'll be so close you can't resist. Tell me your needs by charging, please. The lightning strikes, now make a wish.
Ever wonder who's behind all this? Full juices, milk products, and any sweet mix. Swim, time, and their sugar addiction. Their common desire to surpass constriction. Indiscriminate beings, eating raw and never fleeing. Even human flesh is valid source of food. Oxide waste are fuel, sugar crystal, fable jewel, fellow yeast multiply as the virus per group. Once we sense a tingling feeling, question the truth of what you are seeking. Look at them, their doubling grows. What an army quickly arose. Permeability melts walls. Give or take, unlocking doors. Leaking juice, absorb more booze. Intracellular space turns to ooze.
Even though you cannot see me, I'll be so close you can't resist. Tell me your needs by charging, please. The lightning strikes, now make a wish. Even though you cannot see me, I'll be so close you can't resist. Tell me your needs by charging, please. The lightning strikes, now make a wish.
Strong selective pulses deadly, target cells in any medley. Fix the yeast goodbye with no pain, while bacteria prevail. Post-directic field coat membranes, only program cell death remains. Take from all food in whatever, free tomorrow being clever.
Even though you cannot see me, I'll be so close you can't resist. Tell me your needs by charging, please. The lightning strikes, now make a wish. Even though you cannot see me, I'll be so close you can't resist. Tell me your needs by charging, please. The lightning strikes, now make a wish. Even though you cannot see me, I'll be so close you can't resist. Tell me your needs by charging, please. The lightning strikes, now make a wish.
So it just was a way to say thank you for all the listeners, and yeah, you saw some part of our lab. Thank you for everything. So Michael.
Thank you very much. It was very nice, juicy end. So thank you. So I have plenty of questions, but I before I start, I would like to give to opportunity to anybody from the audience. If any questions are there, please raise your hand or type to the chat. Or meanwhile, if I don't see any, I'll start. So it's very interesting. So of course, many of these things are related to what we've been doing, and also includes some of our unpublished work, like we all been working on the yeast, and of course, the proteins are our main things here. So I'm wondering about if I may ask I was wondering about the paper mentioned with the BSA starch treatment of PEF. For you actually measure the grafting degree, then the hydrophobicity and zeta potential, so basically the size of the particles and solubility. So I've quite some medical questions there, because I mean we are in some things we are doing we would be interested in similar methods, and also we've been doing the potential measurement in the past on proteins. So for the hydrophobicity, what kind of method did you use to analyze that?
Okay, good question. Yeah, I should let me check. Sorry. Okay, so I switch of presentation. Yeah, for hydrophobicity, I think yeah, I need to check because it's work was done by my PhD student, Tara. So I need to check. Sorry.
No problem.
But it's more or less everything is written in that article. So it's
sure. We'll check it out for sure.
To check out. Yep.
Okay. And for the zeta potential, if maybe it'll be a mathematical question, but but maybe you'll know by heart. So there was seeing that there seems to be an effect of decreasing the effective particle size, right?
Yes.
So that's a very interesting. So, but only for a certain range of field strengths. So why is it like that? Any idea?
Question. Yeah, it's really, it looks like that we need to clarify the treatment condition. It's mean that here it's a problem that when we are working with microseconds, so there is a lot of like not how to say there is a lot of space for or is a problem to clarify the effect. So it's mean there is, I can say the temperature effects. Of course, we cannot measure very precise. From a measurement point of view, it's fine. There is no like several cells to just go up in a cuvette, but still, we don't know how the such kind of biomolecules can be affected by even small increase in temperature. In the same time, as I told you, from a biological point of view, of course, erectile oxygen species, yeah, it's still quite hard. We didn't measure, but as we know from biological treatments of cells, treatment of pulsating field treatment of cells, we definitely see the ROS. So it's mean it could be one of the factor which can cause some let's say not clear results. Because I am agree with you. If such kind of effects depend from energy, let's say. So it could be somehow in a line, but yeah, it's the result of not answered questions, and one of it is why it's not linear.
Yeah, especially if it's like peaking, and basically when I'm seeing, I made a screenshot of that particular slide. So I see there is a certain size, like 120 nanometers, which is like a baseline for from zeta potential, and then for PSA starch complex seems to be smaller, and then it decreases, increases. Maybe this can be still within fluctuations, but then for much higher field strength, doesn't seem to be it's again increasing. So it's a very
it's
very peculiar.
In that table BS, it's a mixture of protein and starch. So in this kind of mixture, maybe the particle side can be smaller, let's say, it's a mixture. But after that, it's going even smaller after treatment, mild treatment. So it's but if you're increasing the pulsating field, its strength, it's going bigger and bigger. So it looks like that maybe such kind of is particles start agglomerate. So it's being
aggregating, maybe,
aggregating in between each error. So yeah, but still it's wide. I don't know.
Okay, I see.
Still, we need to pay some attention.
And there's a question here from the audience, Jaroslav Havlicek from our team. So Jaga, I make you a presenter. You can ask the question. So you unmute yourself.
Hello, can you hear me?
Yeah,
yes,
we can hear you.
Yes, I hear you.
Not seeing you, but we can hear you.
No, I switched it on, but for some reason. Oh, okay. I'm sorry for that. Okay, my question is related to electromagnetic design or design of the chip. So it was hidden. Okay, mechanically. Okay. So yeah, so I saw in your design like 3 pairs of lines in a chip. So I guess it's either way how to intensify the field or maybe make active part of it bigger. So my question is if this was, let's say, first approach, or if you also tried another ways how to do it, which fails, and this method comes out as a best one. So yeah.
Okay, so thank you. Let's make clear. You're asking about the electrodes, yes or not?
Yeah, about yeah.
Yeah, so the electrodes, it's why we start from modeling, because we want before developing the final device, we want to understand how we can cover bigger area of let's say the bed where cells are growing in a chip. In the same time, by using different kind of cell electrode, let's say switching composition, we can explore or to treat the cells which were in a between of these electrodes. So it's like it's quite let's say new idea, but it's not tested yet on real sample.
Okay, thank you.
So by the reason why we use not one plate electrodes, but several of them.
Yeah, thanks.
You're welcome. Thank you. Michael, I don't hear you.
Yeah, thanks. I was muted. There's one more question from Vladimir Perez, so I'll make him a presenter. So just a second. Okay. Was there a question from you, Vladimir, or it was just my? Oh, there is. So let's do that. Make a presenter. Change. Yes, please go ahead. If you unmute yourself, you can hear.
Thank you. So my question is there any difference when you treat your proteins? What kind of ions do you have on your electrolyte? Like monovalent, divalent ions, or is there any difference between, say, sodium, potassium, or say calcium, magnesium?
Okay, thank you for a question. I need to check the composition of a buffer, but it's a buffer. If it's I can check, it's no problem. But it's in the same time you can check the latest article.
Okay, thanks.
Yeah, you will find a composition of buffer.
I think also Vladimir, I think it came to my mind, if you playing around with the buffer composition itself, like if you would add or not add particular ions, if that would affect the outcome. Did you have a look on that, or just that was not your interest?
No.
Okay.
Answer is no. It's not tested.
Okay. And for I know I understand that the food application is always complex because every food is tremendously complex. So I get it. It's too many parameters to actually check at all of them at one time. So and for that I'm curious about the starch sort, because we never work with the starch as a sample, but what we do care, and also what's troubling us is often the purities of the sample. I mean, if you buy the BSA from, I don't know, Sigma or different, so there is something there, but not always the providers. They tell you the suppliers provide you the trace concentrations. I mean, of course, as I said, in food business, it's completely different story of complexity, but in a, let's say, cleaner experiments. So there, did you have a look like, if you're wondering about if the purity of that, like, if you buy BSA from this sample and or other company, do you have an experience that will be like differing a lot, the results, or you didn't test that for that?
Yeah, it's not tested yet. The question is very good, and it's always, but in that particular experiment, we buy from regular vendors. So it's I think it was Sigma or Merck.
Yeah.
So it wasn't like the food stuff from
sure.
But still, yeah, of course, if you are, or somebody else working with food stuff from store or just from his garden, it could be a question.
Of course. For example, for BSA, we teamed up with one of our physical chemist colleagues from other institute, and immediately spotted the interesting thing that if you just dilute BSA in a pure water, the conductivity doesn't correspond to what BSA would be, only the pure BSA, because the BSA has a lot of small mobility, right? It's for quite large molecule. So and also by other experiments indirectly, it looks like there is around 1 micromolar of unknown, and it's from Sigma, of course, but they don't specify which kind of ions. There are obviously cations, because the BSA is negatively charged, so it will be definitely carrying. And also BSA is meant to be a copper carrier, right? Typical, that's a role in the blood. So there might be some iron cations or just other atom cations, probably sodium and others, the common biological salt cations. So this kind of stuff, well, for us, we just seen that it contributes certain effects, but yeah, I'm not sure, particularly when you will be adding the starch. I have no experience with starch, so it also highlights how easy it is to dissolve it and other stuff. These kind of stuff, it's also interesting. So yeah, we realize that sometimes it's even when doing clean experiments, there are some tricks that needs to be done to, of course, to purify the molecules to the level that we want to have it, like distinguish why this thing is happening from the other one. So yeah.
Okay, so thank you for this comment. It could be, we need to keep in mind such kind of things. Yep, thank you.
Okay. Yeah, and at the level of the BSA solutions, like out of the cell, not the cells, because it's also can be complex. Now I'm thinking about the reactive oxygen species and free radicals. So obviously there are some ROS produced, and in this, what do you consider to be the mechanism of production for out of the cells? Like if you have only the BSA or BSA starch, what do you think is the production of how they, like, if there is a particular anode or cathode which produces them, or did you?
Yeah, I think it's electrochemical reaction. It can be mainly used to production of ROS.
Yeah,
and we cannot let's say prevent to do that. Of course, maybe we should. Yeah, it's the reason why we want to go a little bit with short electrical pulses.
Yeah.
To prevent such kind of reaction.
Okay. Yeah. That's it. Definitely interesting. Okay. So yeah, in cells it's more complex. I know that well, if you stress the cell, right, that it itself starts to produce ROS. So that's there are two things in parallel. There will be definitely reactive species production at the electrodes and some in the cells as well. So that's might be the one need to distinguish where the ROS come from. Okay. Yeah. So yeah, definitely there are many open questions, and I understand that this is also a rather new field. I mean, for both of us. So I was asking, like, what are the experience you had so far? But definitely BSA is something you are working daily in the lab with the PEF. So we'll be really glad to share more details and experiences, and maybe share some methods to explore.
Yep. Yeah, would be really nice. And still, I agree with you that the protein and PEF, it's really new for our lab, and there is a lot of questions to be answered. Because
will be
with
exciting
cells, especially with the yeast cells, we have quite like long experience, and there is like new challenges, but especially with proteins and PEF, we need somehow to get bigger sample volume, because as you already know, maybe if you we cannot deal, but still if you want to see some effects in a cellular level, for us doesn't enough to play with one cell. We need to play with some population of cells.
Right.
In our like imagination and ideas, it's the same with the proteins. Of course, you can play with one protein, but if you want to see some interaction, still it should be a population of it. It's not only one, not only two. It's much bigger. So in that case, I think the treatment chamber with the flow microfluidic chamber can help us to get bigger sample volume.
I see. Okay, great. So thanks a lot. Definitely there will be many more questions to come, but I think it'll be definitely more effective once we come in person, so we can chat.
Yeah, great.
Okay, so with this, I would like to thank all attendees, particularly to our speaker. And I will stop the recording now.